Okay, good afternoon, everybody. My name is Silvina Horovitz. I'm a staff scientist in the Human Motor Control section, and I will be talking about neuroimaging of degenerative diseases and using Parkinson as an example. So I will be going over these four items, these four aims that we would have when we study any disease and try to show why or how we can use neuroimaging for it. So Parkinson's disease, for those of you that are not familiar with, belongs to a group of conditions that are part of motor system disorders. They are a result of loss of dopamine in the brain, particular in starting from the midbrain and substantia nigra. So the symptoms of Parkinson is defined as a motor disorder because the cardinal symptoms involve tremor, rigidity, bradykinesia, and postural instability. And the treatment, and also one of the ways to diagnose it, is a response to dopamine replacement. There are some problems with that, and on time, or in some particular cases, stop being effective, and the alternate treatment is deep brain stimulation, and we are going to talk a little bit about this also later. However, the disorder has a lot of symptoms that are not related to the motor system. There are sensory alterations. There is, well, voice, speech, and swallowing could be some motor, but are also very common. Gastrointestinal problems, autonomic dysfunction, behavioral within Parkinson's. You can have perfectly normal cognitive subjects, or you can have subjects with dementia and anything in between. There is a lot of psychiatric comorbidities as load of the disease or as a cofactor. So it's a very complex disorder. Now, if we look at what is behind these symptoms, we can think of comparisons with other diseases as well, that you can study each of these symptoms in other cases. But in particular, in Parkinson's, when we think about these symptoms that I just mentioned, are the clinical manifestations, and what we have behind are, of course, some genetic factors. Though not every patient have, at least that we know of, genetic case. A lot of are idiopathic. There are some problems we know with the protein misfolding and accumulation, and we have presence of toxins, right? And we can think of these that will produce some mitochondrial dysfunction. There will be inflammation processes and neural dysfunction as degeneration, and all this together is what will lead to the different symptoms. Now, where in these there is a problem, it's something that some things we can look at with imaging and a lot not. So there is different avenues to study how this disorder comes about and what are the possible modifiers for it. But in the imaging, there are things that we can study and in general are related to these factors. So we can study inflammatory process, we can try to study mitochondrial function, and we can study neural function and degeneration. This is probably one of the most common things that we study, how the system connect to each other. Probably is what you hear the most through this course about a lot of fMRI and some of the structural data. So these different aspects we can study here. So when we look at the inflammatory process and the structure and the degeneration, for example, with the tissue, neural loss will start having more free water, if we think from the viewpoint of the diffusion data. If you look at any other type of imaging, anywhere when we have tissue loss, we will have more partial voluming, probably. So there are signals that will be affected in our part. If we talk about the neurodegeneration and dysfunction, we can study with morphometry studies, just see how the tissue that we can detect, the different contours that we can use for the images change through diffusion, through GRE susceptibility images, morphometry, just the shapes and structures, and of course with fMRI. And we can study, for example, mitochondrial dysfunction with phosphorus 31 spectroscopy. Just as a side note, we are doing here at NIH studies related to all of these, but I'm not going to be talking about our studies. A lot are in progress, and I want to give you things that are already completed and published. But there are all different aspects that can be studied. And of course, we want to see how these different parameters relate to the symptoms. We know that they are there, right? So as I mentioned, the structural changes could be seen through T1 weighted images, diffusion, susceptibility, neuromelanin. Metabolic can be seen through spectroscopy or PET or SPECT, and functional changes through functional MRI, both task and steady state. And the question is, first, what changes between the populations, but also whether these changes are related with any of the clinical or electrophysiological measures that we can take to the patients, trying to understand what is the meaning of a change that we can see. So as I mentioned before, we can use imaging to try to understand what's going on in these diseases. For example, identify the changes, though we need to define whether the changes are what are causing the disease or are a consequence of the disease progression or the treatment that we are using. We would like to know, for example, if something would predict the onset of a disorder if we have a population at risk. We would like to do, as I said before, differential diagnosis. We want to understand how these metrics, as I said, correlates with the clinical outcome, so we can see the disease progression, and then we would be able to evaluate if some disease modifier is useful or not, because we need to know what is the progression to see whether we can slow down. We need to know what is the ultimate goal will be to avoid someone to have the disease, right? So we need to be able to understand what will generate this, what will be the onset and before the onset of the symptoms that we usually characterize a disorder, whether we can get that. So for doing all that, we need a large population. As I mentioned before, there is all these cofactors for the disease, so it's hard to get an homogeneous population, or if you get an homogeneous population, you are neglecting a lot of the patients that have these. So if you want to include more covariates in any clinical study, you need to have larger populations. Of course, that we need to see how people change over time. That's harder, takes time, so also we need larger cross-sectional studies. We want to see the populations at risk, and we want to do populations with similar manifestations if we would have wanted to get some sort of biomarker for the disease. So how we are going to study each of the changes or the different differential diagnosis treatments depends on the question, and we will need to figure out what modality of the images that I mentioned before will yield some information. So as I mentioned before, we also need to, I think I missed to mention that we also want to compare what is the normal, what are the normative values of whatever we are measuring, and whether those changes, for example, with age. Talking about that, Parkinson's disease is usually a disorder that affects elderly population. So when we do imaging, we need to deal with other cofactors. For example, the brain atrophy. We are looking at the neurodegeneration. That is what we are trying to study, but also will affect, for example, how we normalize our data, what are the differences that we are checking, right? The patients, as they get older, having Parkinson's or not, will have more white matter diseases, will have some findings that are not clinically significant, but still would affect the images, and one need to take into account. Into more practical terms, when we deal with Parkinson patients, as I mentioned, the typical features include rigidity, and also they have tremor and dyskinesia. So all these would affect the rigidity, for example, how you put the head of the subject in the scanner and how can you move it, or the tremor, whether the amount of movement, the motion is similar or not. Dyskinesia is where they will have sudden movements in the scanner. So those things I need to take into account when you are looking at the data. So just getting more on practical terms of what I'm going to be, the studies that I'm going to be mentioning today. So to talk about the pathophysiology of the disease, I would tell you about one of our fMRI studies. I will mention some of the morphometry studies. We look at disease progression through longitudinal studies, some differential diagnosis. I'll mention some MRS spectroscopy data and how we assess treatment. For example, how we have been using DBS, DTI tools for DTI tools to understand how DBS work. So as you see, there are a lot of modalities and a lot of studies. I will just briefly mention some, go more in detail into others. So the first study that I want to tell you is this, is one that we did here a few years ago on understanding some of the functional networks in resting state in fMRI. This is just a schematic of the typical systems that we think of when we, the brain areas that we think of when we look at Parkinson's. Just want to remind you, when we look at the cortex here, we have each in parallel it's not only the motor, but also we have all the cognitive, executive, and different subsystems that share these pathways. So one of the problems that Parkinson patients have is that things become smaller, what is called sequence effect. For example, if someone asks them to tap, they will tap like this and eventually will be this. If they write, the letters will be normal and eventually will be smaller and happen in different things. So we wanted to study what is this, right? What is the difficulties that these patients have are in self-initiating a task, but also in maintaining a task at a steady level. So we know that there are some motor deficits in them. However, when you ask the patient to follow you, if they have the visual feedback, they are able to perform this task. So we decided to study this on resting state with using some graph theory analysis. So I'm not going to get into detail about all the metrics that one look in network analysis. I don't know if someone else have give a lecture. If not, we can talk in detail about this. But the idea is that we're looking at the connectivity, but instead of looking at the connectivity between one RI and another, or between one node and another, one voxel and another, we are just as in a pair-based comparisons, we are trying to understand some metrics that involve how that particular region or voxel is affected by the connections of everything with everything, right? So just integrating the connectivity in a way. And there are different metrics that one can tell that are listed in there. So the model that we tried to study was, well, we have the sensory motor network that is the one that will execute this open and closing of the finger, right? But also we need to get some network that will keep the task going. And so we selected the singular network. That's a task set maintenance that once a task starts, like it keeps the mental state maintained continuously while you're doing it. And we know that the frontoparietal networks would affect the adaptive control. And we know that this task is affected whether you have feedback or not. So we just decided to look at these particular areas on a group of 30 healthy volunteers and 30 patients of medication. And these are the matrices of correlation. So this is what is called the adjacency matrix, is the voxel region by region correlation. But then we can apply some tools and extract the parameters and trying to figure out, we could have done like just a t-test between these two, right? But it's not telling much beyond a single connection. So what we look is at a little bit more of the parameters. So that we look at the cluster, north strength will be the connection of one voxel with every other voxel. We look at the clustering, so how things clump together. We are looking at the local efficiency, so how good the connections are in the local environment, and the between the centrality, that is how how central is an area in connecting the others. And we see that definitely the sensory motor area is affected in the patients, but also there is the singular network had a lot of areas that are affected. And in particular, we see here the TPJ and the insula that are affected. So these areas are of interest because are related with self-awareness, and we try to look at the difference. In particular here, explore a little bit more, and as you can see, there is a big difference that the patients start losing these connections, and they have a much thinner overall network with the TPJ and the dorsal anterior insula. So definitely the deficits that they have, even though their motor, are caused probably more for deficits in other areas of the brain that usually we don't focus that much on Parkinson's. So of course this is an analysis that we did on a particular group of brain areas with a very clear hypothesis on where we think the deficits could be, and we find them. And well, this could have implications, for example, into how one can try to work with the patient for rehabilitation. If we know that these aspects will affect eventually their motor behavior, then we can work on strategies that will bring more their self-awareness. We know that in Parkinson's, but now we can see it through the brain function, that is not just happening that they're more aware. There are these networks that engage differently. So if we bring back these networks of self-awareness, we can get the subjects maybe to perform better. So in that particular case, we did an analysis only in particular brain areas. And this is a problem that we have in general when we look at the brain or study disorders. You can look at the brain based on a hypothesis and just pick the areas of the brain that you are interested, and you are more likely to find a response that you will be able to interpret because you know what are the components that you are including in there and why, or what will be the role of each of them. Or you can try to explore the whole brain. So we try to do that as well. This is in a different group of patients, and these are on medication, so it's completely different group of subjects. But we try to look at the whole brain. And this is, for example, we use the Craddock 200 to divide up the brain. And this is one of the problems, right? When you divide the brain and try to include the whole brain in the analysis, then the parcellation of the areas not necessarily have the biological meaning or the functions that, the way that these parcels are divided, not necessarily match with the functions that we know, right? These try to be the more consistent, and there are different groups using different number of regions. Whatever you choose, still you will have the same problem. You won't do this on a single voxel level, and then is how much these are reflecting the real functions. But anyways, you can still do this, and when you put the whole brain, what we try to see is, for example, how modular is the brain. So how we can divide up all these regions into groups, how they come up together. And what we see is that there are mostly, we can find similar things in the patients compared to controls. However, the healthy controls have some basal ganglia clusters, models that we don't see separated in the patients. Some of the frontotemporal areas and the nodes in the orbitofrontal cortex come different. So this also tells us in general which areas are more affected because they just lose their I wouldn't be the right word, in the independence, right? They start kind of melting with the others. You don't get that specificity. That's things that I have been seeing in particular areas. For example, in patients, there are studies that have shown that patients lose specificity in the connectivity of the, for example, the putamen, that you don't get the differentiation of the anterior and posterior putamen as good as you can find in the healthy controls. So these, as I mentioned, so we see that this lack of molar distinction, in particular in the orbitofrontal cortex and the basal ganglia, could be related to things that we see in the patients. We have alteration of modularity as well in some core functions that will affect any task, but also in the sensory motor networks. I will show, this is, we suspect that the dopamine treatment won't improve all the networks the same way. The contrary, they will be disruptive in some networks. That's why we could see some networks that are not affected. That's our speculation, right? There are networks that are not necessarily affected by the dopamine. We know that these patients have dopamine. However, we do find that the networks are affected. And indeed, there is another group, Brian Berman, that used to work with us. I have done this comparison on the modularities. It's slightly different because he didn't study the whole brain or particular regions. He divided up in networks. But still, we can see that there are differences between healthy controls and Parkinson patients off medication. And when we get on medication, you have probably even more differences than off medication. So while we are correcting some of the networks, still the medication is affecting other networks. And this is something that we're seeing in our data on and off. I have seen, so it's kind of counterintuitive, but I have seen several. And the reason is that if we look at some of the parameters, I don't know, I don't know if you can see my labels there are too small, but this one that is the most different at the level of both whole brain, local and global efficiency, the smaller and the most different is the patients on medication. So we are regularizing some of the metrics, but we also affecting others, and that's something to explore more definitely. So besides that, the things that are ready to be explored more, and how the whole brain is affected by the medication, even though we know that there are benefits for the patient. Of course, there are a few questions to ask. One is for the whole brain parcellation, as I mentioned before, how we define the nodes and how we study the different parts. And in general, when we look at, just remember these two studies are resting state. It's not the same that study the subjects during tasks. So it could be that we are affecting these networks at rest, but they definitely would be working better, or maybe working better during tasks. There are questions how these metrics correlate to clinic, and that's a very different question because, again, there are so many parameters that one can correlate with the clinical aspect of the patient that we'll need larger and larger populations to get that. How much rest is rest? As I mentioned before, the patients may have tremor, may have dyskinesias. Healthy volunteers, these are not the professional 20-year-old volunteer in the scanner. These are people that match in age our patients. So the patients in most of these, our studies and other studies go probably the youngest will be in the mid-40s if we get any early Parkinsonian patient, but mostly will go to their late 50s and most likely in their 60s or even 70s. So it's not the same than a younger person being in the scanner for either patients or healthy volunteers. In terms of how much motion or active motion suppression is there is something hard to measure. What we do is usually try to measure some properties of the fluctuations or the signals in the motor cortex, besides looking at the motion parameters, but we try to compare the properties of the motor areas and see whether they're equivalent. We can assume that they're acting in similar way. Of course, like in any resting state, what is the alertness level in the subject? It would be a limitation. There are ways that we have done on looking, for example, the fluctuations in visual cortex or things like that to try to establish the overall level of the patient. And of course, the question when we see differences based on these is that we are looking at a state or we are looking at a trait in the patient. So just this will be my last slide on the fMRI part, and then we move to more structural data. So we know that Parkinson's disease affect the networks, brain networks beyond the motor system. The connectivity studies can help in trying to identify what is the complex, some of the issues or the parameters in this disorder. We need to take into account that is when we are talking about rest, as I mentioned, the actual limitations that the study have. We know that there are deficits that are related to disease progression, and there are things that will be affected by medication, and we need to separate what is the cause of the consequence of this, if we could. And then we want to see, it will be good to see whether the functional changes are preceding or preceding structural changes, for example, in tractography. So, and then the second question, if we are doing this type of studies, is whether these limitations are due to the progression of the disease or limitation in the methods. Just we need to remember when we do a study, especially structural DTI, like if we don't see something, doesn't mean that it's not there, right? We need to be aware of what are the limitations in the methods that we are using. So I want to switch now a little bit on some of the structural data that are being studied for understanding Parkinson's disease. I'm not going to get into the details of each of the imaging modalities that I mentioned. I think are covered in some of the other lectures. But as I mentioned at the beginning, Parkinson's disease is neurodegenerative disorder, progressive neurodegenerative disorder. And one of the characteristics is the dopaminergic cell loss in the substantia nigra. This is some histological images. And what I wanted to point here is that depending on what you're trying to trace, you can define different parts of the substantia nigra. So this is iron content, and this is neuromelanin, for example. So when one tried to study this region, one of the things is how we are going to define the region of interest. That's very, very critical, and I think is a limitation on most of the these morphometric studies that try to find whether one with MRI can detect these changes and progression of changes in cellular loss, dopaminergic cell loss in the substantia nigra. So the things that people have used is looking at different shapes and different contrast, either in longitudinal studies or cross-sectional studies or a combination. So none of these methods or these examples that I'm going to show is the gold standard. Still, we don't have a gold standard. I just wanted to show you how different groups have approached and what are the things that people are looking, and try to think what are the limitations on them. So one of the studies, a couple of studies from, this is 4 years ago, for example, and I think there were some studies even before this, but what is calling this swallowtail. So this definition, sorry, here, of the shape of the substantia nigra, where there are groups that define that on healthy subjects, there is this indentation in the tail of the substantia nigra, and the claim is that that tail, that shape is lost in the patients. So so there are a couple of groups, at least, that have shown that and reproduce it and consistently or mostly consistently find these in the patients and not find it in the find it in the healthy volunteers, the indentation, but don't find that in the patients. However, it's it depends on probably on how the images are taken, and indeed it depends on the quantification on how that is drawn, right? If you look, for example, these images is from another group that they tried to define the boundaries of the substantia nigra. Now, you don't see that tail anymore, right, that you would see here, where here you don't see the boundaries very well. So the contrast and what you're trying to get will make that you see something or not, and how you try to quantify, for example, the volume of the substantia nigra or the shape would depend on what approach you're taking, and which one is the right one is hard to define. So this is also data that has several years. This is from a group in Korea, and this is from the same group where they consistently find that the shape of the substantia nigra lose their smoothness in the side on the patients. This in the 7 Tesla with GRE, for example, we can see, we haven't quantified in our data, but you could see one of the problems with these, as you see, I mean, these are beautiful images, but we know that when we have a subject in the scanner, you don't get these images in everybody. These are areas that are very affected by the respiration, by susceptibility, by motion. It's not a quality of image that you get in every single subject, though you should aim for that. So some of the other, as I mentioned, other of the parameters would be the neuromelanin. That would be kind of the opposite side of the substantia nigra, and probably could be what they're defining in the first study that I show you as a little dip in the tail. There will be, for example, this part. So they define 5 different aspects of the negrosomes where they can detect the neuromelanin, and this group claimed that there are differentiation, and you can separate clearly in the neurosome. I don't know why we are losing the image there, but you can detect here how controls have a much higher value, and the signal keep on changing, I mean, losing as the disease progression. The UPDRS is the a measure of the disease progression, and so this will be a very light level, assuming that this is of medication. But this will increase. However, for example, if we look at this, the patient on medication will have a different scale, and probably this won't change. So there are some factors, but yeah, according to these, you can detect in these little areas based on the intensity how much progression there is in the disease. The question is, how do you draw these areas, right? How good are you at drawing this area, and how these will affect your results? So this is a complex question, and even though these studies claim that the iron-rich areas are not given the information, actually, if you look at here, still you have a gradient from healthy volunteers to patients. The question is how good we could be at separating this. And I think what is more critical is that if we try to use this for individual subject data, that all these have some sort of overlappings. So you can't, even though at the group level you have significant differences at the individual subject, still none of these metrics could provide a good separation or a good diagnostic tool alone. So maybe diagnostic tools should come from combination of different metrics, but definitely none of these will act as a biomarker. Other way to look at the data is, for example, look at the diffusion data, the structure of the white matter. And there are many studies that have looked at different parameters. This particular study is just published. And what they do, this is the reason that I'm showing is because it's a longitudinal study, so they have two time points on each subject, and they have healthy volunteers and patients, kind of similar to what we are studying now, but we haven't got to the point of collecting our second data set completely yet. But so if you have the same subjects coming twice, and the healthy volunteers and the patients, then you can see whether the evolution or the changes in the patient go at a different rate than the changes in the healthy volunteers. So there are a few areas that that you can see changes here, and the patients they have deficits faster than the controls in the midbrain. At the same time that the FA decrease, the MD increase in the same area. There are so this allows to see where the regions are progressing into the disease differently. There are some limitations, and of course, the question is the registrations. And I mean, again, I'm not going to get into the specifics of the methods. I guess Joel and Carlo will talk about the optimal way to collect and process the diffusion data. So I just want to say that all the studies that I'm showing, including ours, right, will have limitations, but these are ways that one can go around. So there is one group, this is one of the papers from the University of Florida, that had been pushing into looking at the free water in the diffusion data. So when you get your tensor in the DTI, if you have more free water, your Phe values will be different. So they propose a model that is very simplified and claim that there are group differences in the posterior part of the substantia nigra that would be this dot here. So one of the things is that they look together at both hemispheres. The other is that even though the difference seems significant here, when you look at the actual data, again, there is a lot of overlap in the patients and the healthy volunteer. This is a correlation with the MOCA, that is a cognitive test, very comprehensive and not necessarily would be related to the substantia nigra, but will be more a general cognitive test. However, again, even though they have these differences here, just there are a lot of limitations in the way that they model the data, because they use some standard values to normalize the whole brain, and it's not really a proper estimation of the tensors. So, the last, well, no, two parts. So the MRS part that I mentioned, so differential diagnosis. We can use imaging also for differential diagnosis, and I think this is one of the examples that have been more concrete and more standard in one of the few cases that you can say, well, it has been distinguished, and if you try to do it, even you can get a sense of that as well. That is based on the multiple system atrophies are at the beginning very similar to Parkinson's, and sometimes may be hard to do diagnosis. I mean, in some cases, diagnosis, the patients do not respond to dopamine treatment, and then you know that more likely is MSA. But in some cases, may respond a little bit, and it may be confusing. And sorry, I don't understand it. And so there have been for many years now studies of trying to use magnetic resonance spectroscopy to find differences. And there are in proton spectroscopy, there haven't been differences in the Parkinson patients. However, different brain areas, for example, in the putamen, they have seen a difference or a trending differences in the progressive supranuclear palsy, and in some cerebellar areas in the multiple system atrophy with the cerebellar characteristics. That's more of ataxias. There are also differences, very clear differences, again, in the MSC when you separate that particular type of disorder. However, if you mix them all, you can't. And this is one of the few, one of the few cases where you can see in this brain area that there really a separation between the different patients. So that could be something more useful if you have enough normative data on healthy volunteers and PD patients, for example, that will be most in this area. I mean, it's not 100% separation, but definitely it goes to a different area that will allow for classification. So going to the last part of the talk, I mentioned that we could use imaging as well to do treatment assessment. And I mentioned a little bit about on and off medication studies. I'm not going to go more in detail into that. I mean, I'm not going to show you any particular study, but that will be similar to any other pharmacological study, though we are not looking at the onset of the response, but rather you can do any fMRI study that you typically do, but then when we do those studies, we perform the study on and off medication and compare the healthy controls to the patients in one condition and then the patients within themselves in the other condition. Of course, you need to think about the statistics, because in one group, I mean, you can do two measures to look at variability, but still is one only state, because the healthy volunteers do not have the treatment, and it just, I mean, there are some studies that have done dopamine challenges or dopamine given to the healthy volunteers and then compare, but really when you don't have the deficit is a different response than when you do, so it won't be a fair comparison. And beyond the getting the IRB approval to give those medications to the healthy controls, but then you do a comparison between the patients and the healthy volunteers in one of the states and between patients, and then you can try to figure out what is whether the medication is, for example, restoring the lack of function or affecting some other brain areas. And with DBS, there is different things. The brain stimulation, as I mentioned, is one of the techniques that is used as a treatment when the patient stop being responsive to treatment. The surgically invasively, they can be stimulated. There can be electrode put inside their brain, and that would aim at improving their quality of life. I would mostly remove the dyskinesias and the off-medication on-off cycles that they have, so keep them in a more stable condition. There are, I guess I would say, two different approaches to look at the assessment of the DBS. What I'm showing here is the study done by Horn, and it's a group of Michael Fox in Harvard. And they base their studies on data from the databases. So they use the Human Connectome project for the diffusion data and for the resting state. And mainly they get the location of their electrode, normalize that to standard space, and look at where they're applying the stimulation and where the connectivity of these areas, and try to evaluate where the results, and if that case didn't work, where these connections were going. One of the limitations is that if you use this, you're always going to the mean and the standard subject, right? So you're losing the specificity of the subject. I'm just putting here the databases that they're using. They have not only the Human Connectome project, but also you can use the PPMI is a database available on Parkinson patients, and it's open for researchers, and you can get different type of information, different populations to study. So our approach for this is slightly different. We use the actual data from the subjects, and we use the tractography from each individual subject. So once we put the once the electrode is in the brain, we can find exactly through registrations of the different images that are used for the surgery where the contact is, and we can use that information to do tractography on the diffusion data collected before surgery and try to figure out where the stimulator is stimulating based on the actual location and the actual voltage of the stimulation. So the difference between the method that I showed before and our method is that we use the actual subject data. So if we are taking into account the subject variability. And what we can do is getting the different voltage, like whether we start having a response, a standard value, or whether we have side effects. And then we can see that these tractography systems are quite different. So these allow us to start analyzing either whether a particular simulation will be beneficial, or start learning in general where what type of connections are beneficial for the subject, but just by knowing the actual data from each one. And just the last example, For example, here this is one of the data sets from the same data set, but now we can see, for example, before we can say, well, our stimulator goes to the thalamus, but now we can also say where in the thalamus and which from the connectors are the subjects that have a good response to that particular contact and that particular voltage. Where do we go in the thalamus? And indeed, for example, if we go to the motor nuclei of the thalamus, we get a good response to the treatment. So this would allow us to start doing some type of inferences on what is that we are obtaining, or what's the best way to treat the patient. So just to conclude, I try to show you different image modalities that we can use to look at different aspects. There is a lot still to do to understand the disorder. As I mentioned, the complexity of all these neurodegenerative diseases make that it's hard to take into account. I mean, you see that I barely mention correlation with the clinical factors, and that's something that is really important, because if you have a Parkinson patient and you see it walking on the street and you're trained, probably you will see, or you will guess that the particular person may have Parkinson, or at least a Parkinsonism. Yet, if we put them in the scanner, it's very hard to find the differences. So until we get to be able to match or be better than the clinician, we are still limited in what we can do. But there are progresses, and there are a lot of things that we are learning. So there are a lot of work to keep doing. Thank you. So I'm open for questions if anybody has.
A lot of your resting state data is based on populations where you see changes in global connectivity. And so what have you done to try and look at some of the networks in individuals longitudinally?
We are still, in our case, we are still collecting the data, the longitudinal data. So you'll need to wait. Probably we'll having most of the patients coming back within this year. So next year I would be able to tell you in particular. But the things that, so there are two things that we are doing. One is to make sure that the changes, I mean, there are a lot of changes, I think, so even though we have a lot of stability in the resting state, you may have a 30% of variability even within a day or not. So we are doing, in one of the sessions, we are doing a second repeat measure. So we'll have two measures of the same subject, and we are doing the same on the healthy volunteers, and we're also having the healthy volunteers coming twice. So ideally, we would look at, we can see, so we don't have, in these patients, a lot of the cognitive or a lot of the task related, and that's something that we may want to change in the future. But there are some metrics that we have from the MMSE or the MOCA, whatever test they take. So we can get, or the motor system from the UPDRS. So there are some metrics that we can take that will be different between the subject, right, between visit 1 and visit 2. And we try to look not in general at the whole brain, but really at network specific related to some metric that we can have, and we can evaluate whether there is a change in that, or because otherwise we are... I don't know if that answered, but yeah. Yes. So yeah, that's what I was saying. Like, if we want to look at, well, definitely we're going to look at the motor areas, because we definitely have motor scores. So yeah, we can start from a seed in the motor cortex and follow through. But that's why I'm saying we can, if we look at the executive, we can look at, a brain area that, depending on the task that we are looking, right? So if we, for example, we're looking, as I mentioned before, at the sequence effect, and we have something on their ability to maintain a task, we can look at the anterior insula as the seed to start. If you're looking at the verbal fluency, we can look at the seed in the verbal fluency. So that's the way. But we can do it on the networks that we have some cognitive task. And we can do that both in the functional, and we are also collecting structural data. So I think beyond looking, I think the most interesting part will be beyond the individual seed connectivity, is looking eventually whether morphometric diffusion or functional changes are coming first and how they relate to each other, probably. Any other question? Okay, thank you.